Welcome to a video about spike proteins, where we're going to talk about spike proteins. But Paul, you ask, what are spike proteins? Good question, it's almost like there's a script or something. Cornell University describes spike proteins as enveloped viruses access their host cells by binding to receptors in the plasma membrane and then undergoing fusion with the host membrane. Both binding and fusion are mediated by a specific viral spike protein that is typically primed for fusion activation by proteolytic cleavage to expose the fusion peptide. But that's kind of long and it goes on like that for a while, so I'm just going to try to simplify so it can make more sense. Basically, spike proteins are responsible for a virus being able to infect a cell. The virus goes up to a cell and the spike proteins are what allow it to get through the outer membrane. Then once it's inside, it can start replicating itself and spreading the disease. It's quite rude if you ask me. In structure, it's kind of like a burr, the kind you find at the edge of a public park when you accidentally kick the ball over and then you gotta run all the way over and get it. Yeah, kind of like that. But in function, it differs a lot. See, a burr just hooks on to be moved around. A virus hooks on in order to be able to get into the cell and then take control and replicate itself. So usually our immune system is pretty good at counteracting these viruses, often by taking out the spike proteins on the protein surface, which then make it so the virus can't hook on and get in and create more of itself. But this isn't always possible. Normally, the way our immune system can detect these foreign proteins is via amino acid sequences, but sometimes the pathogen will put sugar on the protein surface, which quite easily hides it from our antibodies. Many viruses do this, like influenza, or who. Specifically, the virus, once it's inside your body, finds a single receptor molecule and attaches to it with a spike protein. Then, it's able to get brought through the outer membrane and into the cell, kind of like unlocking a door with a key. Once in, the virus can get to the genetic material inside the cell or the nucleus, and then it can take control and start replicating itself. Well, I guess it's time to talk about COVID-19 now, so I'm going to put on rubber gloves just to be safe. And for dramatic effect, I guess. But Paul, you ask, how does the topic of spike proteins apply to the coronavirus? Great question. It really does seem like there's a script or something. So the coronavirus, as you would expect, has spike proteins that it uses to grab onto the cell and then get in. SARS-CoV-2, the virus, uses spike proteins to get COVID-19, the disease, into the cell. In fact, spike proteins in the coronavirus, which make up this little halo type of thing, are actually the reason that it's called the coronavirus. Corona meaning crown in Latin, and this kind of looking like a crown. Once the COVID spike proteins have locked onto a receptor molecule and been let inside the cell, they can access the host's ribosomes and begin creating more copies of themselves. Again, it's quite rude. Spike proteins are really important, obviously for the viruses themselves, because it's what they use to get into the cell, but maybe even more so for us. If we could successfully find a way to stop spike proteins from fusing with the cell, such as through the use of antiviral peptides, we could theoretically stop SARS-CoV-2 from being able to spread. It could also possibly be used in the treatment of those who are already infected. Scientists around the world are working frantically to find a cure for COVID-19, and spike proteins hold a major source of research, and maybe even possible solutions. But for the moment, we're just going to have to wait and see what they can come up with. So I wasn't exactly sure what to do with all my sources, so I just made this nice little end screen, and I guess I'll just stand here. I'm not wearing a watch. Okay, well that's the end of the video. Thanks, bye.